In this presentation, we are going to discuss retinal venous pressure RVP. It is an exciting and clinically very important topic. We are interested in the pressure in these veins here in the retina, and we will discuss the following questions. Why is it important? How can we measure it? How high is it? What influences it? Can we lower it? When a patient is coming to see me as an ophthalmologist, I focus on different aspects of ocular circulation depending on the patient's disease. We now focus on vascular dysregulation and in particular on dysregulation of the veins in the retina. Blood vessels are not passive tubes. They are all actively regulated at any time and place. This also applies to the veins in the retina here. Why do we need regulation? In order for the eye to function properly, the blood flow must be adapted to the actual needs and be as independent as possible of other factors such as fluctuation of blood pressure or intraocular pressure, physical activity, ambient temperature, or mechanical forces, and body position. Because this regulation is so important, many mechanisms are involved. I have outlined some of the most important players here. The blood flow in the retina is influenced, among other things, by its current activity. This is called neurovascular coupling. While the blood vessels in the retina have no innervation, the vessels of the choroid are intensively innervated. This is important for thermoregulation, for instance. The situation in the optic nerve head is special, because here the vasoactive substances of the blood can bypass the blood-brain barrier. They diffuse from the fenestrated capillaries of the choroid into the optic nerve head and adjacent retina. But common to all blood vessels is the involvement of vascular endothelial cells in regulation. The vascular endothelial cells line all blood vessels inside. They have a number of functions, one of which is the regulation of vascular tone. The endothelial cells, here in blue, receive physical and chemical information and in turn release molecules that influence the tension of the smooth muscle cells, here in red. One of these molecules is ET1, endothelin-1, a vasoconstrictor. It is a peptide of 21 amino acids with a half-life of about 15 minutes. And the opponent is nitric oxide, NO, a vasodilator. It is a very small molecule that easily diffuses, but has a half-life of only about 10 seconds. In our body, we have roughly 10 trillion of such very active vascular endothelial cells. Back to our veins. When we observe the blood vessels at the back of the eye, we see that in most healthy people, the veins in and around the optic nerve head pulsate. The reason for this pulsation is by no means trivial. Many scientists have tried to explain this phenomenon. A good explanation, together with the corresponding literature, I have summarized here. In our present context, however, more important than the cause of the pulsation is the fact that the pulsation tells us that the pressure in the pulsating vein roughly corresponds to the IOP. But what is a normal retinal venous pressure? The pressure in these veins is at least as high as the intraocular pressure, otherwise the veins here in the retina would collapse. It is also at least as high as the intracranial pressure. This because the central retinal vein leaves the eye via the optic nerve. And finally, it is at least as high as the pressure in the orbital veins, because otherwise the blood could not flow. In most patients, however, both the intracranial pressure and the pressure in the orbital veins are lower than the intraocular pressure. Therefore, the question normally is whether retinal venous pressure is equal to or higher than the IOP. This brings us to the question of how we measure RVP. As we have already noticed, if a vein pulsates spontaneously, we know the pressure in that particular vein is equal to the IOP. If, however, there is no pulsation, we can increase the intraocular pressure until we see a pulsation. This artificial IOP then corresponds to the pressure in that vein. We can raise IOP either with an ophthalmodynamometer or with the IOP stim. How to do this exactly will be explained after my presentation by Mr. Kraus from Emer. But what happens if the retinal venous pressure is increased? This picture here symbolizes the blood flow in the retina. Here at the entrance to the eye we have the arterial pressure. And here at the exit of the eye, the venous pressure. The difference is called perfusion pressure, and the resulting pressure gradient drives the blood flow. Now, if retinal venous pressure increases, perfusion pressure decreases, and the pressure gradient gets flatter. If there is no sufficient contrarregulation, blood flow decreases and the risk of hypoxia increases. But the high retinal venous pressure has additional consequences. The pressure in the blood vessels is normally greater than the pressure in the surrounding tissue. The resulting transmural pressure pushes fluid from the capillaries into the tissue. A part of this fluid is reabsorbed by osmotic attraction, but another part flows off through the lymphatic system. The retina, however, has no lymphatic vessels, but does have blood-brain barrier that allows a stronger colloid osmotic suction. If the retinal venous pressure increases, the transmural pressure of the capillaries also increases. This eventually can disturb the physiological balance and contribute to edema formation. In many diseases, retinal venous pressure can be higher than IOP, for example in glaucoma. Pilanat et al. Observed that the difference between IOP and RVP is particularly large if glaucoma damage has reached a certain degree of severity. The resulting decrease in perfusion pressure is particularly relevant for glaucoma, because a low perfusion pressure is a known risk factor for progression of glaucomatous neuropathy. Since it was previously assumed that the retinal venous pressure simply corresponds to the IOP, perfusion pressure was defined as arterial pressure minus IOP. But the retinal venous pressure can also influence perfusion pressure in the optic nerve head. While the arterial supply comes from the ciliary circulation, the venous drainage is through the central retinal vein. If the pressure increases also in this part of the vein, the perfusion pressure in the optic nerve head drops. Therefore, we defined perfusion pressure today as arterial blood pressure minus RVP. But what leads to an increased retinal venous pressure? Is it a mechanical compression by the lamina cribrosa or by a stiffened artery? Or is it a consequence of a focal functional vasoconstriction, as we have postulated many years ago in the context of the pathogenesis of retinal vein occlusion? The lamina cribrosa undergoes a significant aging process, which is further accelerated by smoking, but neither age nor smoking affect the retinal venous pressure. In patients with diabetes, retinal venous pressure increases with the occurrence of a retinopathy, here marked in green. Therefore, we asked ourselves whether nitric oxide or endothelin might be involved in the regulation of RVP. When patients with diabetes develop avascular endotheliopathy, endothelin increases and nitric oxide decreases. Now we want to see if this could explain the increase of retinal venous pressure. To study this question, we measured retinal venous pressure under different conditions and observed that in healthy climbers, the RVP increases with height. Michael Bertje, a PhD student at that time, measured IOP and RVP at 300, 4,200 and 6,000 meters above sea level and found a mean increase of about 10 mm mercury. This means a low oxygen partial pressure increases the retinal venous pressure. Why is that? In the previous study, also with climbers, we had shown that the plasma level of endothelin increases at altitude. This can be explained by the factor called HIF-1 alpha. HIF is continuously produced in all cells. At normal oxygen concentration, it is oxidized and then degraded in the proteasomes. If oxygen concentration drops, less is broken down and HIF concentration increases. And because it is a transcription factor, various genes are overexpressed. This includes, for example, EPO, erythropoietin, which stimulates the production of hemoglobin, or VEGF, vascular endothelial growth factor, which is produced, for example, in hypoxic maculas and leads to neovascularization. Less known is the fact that hypoxia also stimulates the expression of endothelin. In patients with high endothelin levels, for example due to autoimmune diseases, we made the clinical observation that the veins in the retina were rather dilated but constricted in the optic nerve head. At lower endothelin levels, this was partially reversible. And we further realized that the pressure in different veins of the same eye were often quite different. You and colleagues then used in vitro studies to show that endothelin actually constricts the retinal veins. But in vivo, endothelin, which circulates in the blood, has little or no effect on the blood vessels in the retina because the blood-retinal barrier prevents direct access to the vascular smooth muscle cells. Endothelin, however, constricts the retinal vein when it can bypass the blood-retinal barrier. This is the case when endothelin diffuses from the fenestrated capillaries into the optic nerve head and the adjacent retina, or when it is produced by a diseased artery and diffuses to a nearby or even crossing vein. From our previous studies on isolated ocular vessels, we know that veins are about 10 times more sensitive to endothelin than arteries. Finally, endothelin can also originate from the adjacent hypoxic tissue. This, for example, is the case in patients with progressive retinal vein occlusion. It is not surprising that the retinal venous pressure is elevated in eyes with retinal vein occlusion. Rather surprising to us, however, was our observation that the retinal venous pressure was also increased in the clinically healthy partner eye. We explained this by the increased endothelin plasma level observed in such patients. Endothelin was elevated even in young RVO patients who had no classical vascular risk factors other than primary vascular dysregulation. According to the classical theory, the cause of retinal vein occlusion is a thrombus. According to our hypothesis, endothelin and other vasoconstrictors can constrict the retinal veins locally, thereby causing increased retinal venous pressure and, in extreme cases, even retinal vein occlusion. Finally, Kita and colleagues have shown very nicely with animal experiments that an intravenous endothelin infusion leads to a constriction of the veins in the optic nerve head. But where does the endothelin from our patients come from? Already under physiological conditions, different cells synthesize endothelin. It is, however, primarily produced by the vascular endothelial cells and mainly secreted abluminally. A small portion is released luminally and influences thereby the plasma level. Under pathological conditions such as hypoxia or inflammation, other cells build endothelin and increase the local concentration, but also indirectly the plasma level. A few general remarks about endothelin. There are several isoforms. We speak here only about the endothelin 1. There are different receptors with different functions. In this context, we are interested in the ETA receptor on the smooth muscle cells. We talk about endothelin plasma levels, but these do not have to be identical to the concentrations in the tissues such as retina or optic nerve head. The quantification of endothelin is not easy. One reason for this is the half-life of only minutes. Besides the endothelin concentration, the endothelin sensitivity is also important, and this sensitivity depends on the receptor density in the corresponding tissue and can also vary individually over time. We have shown in glaucoma patients, for example, that the endothelin sensitivity increases as blood pressure drops. And Chen et al. Have demonstrated that homocysteine upregulates the endothelin A receptor in vascular smooth muscle cells. Now we will briefly come to a special clinical situation, namely the primary vascular dysregulation. If blood vessels are not regulated according to the needs of the tissue, it is called vascular dysregulation. If this is not caused by a disease, but rather by a predisposition, it is called primary vascular dysregulation, PVD. If PVD is combined with certain signs and symptoms demonstrated here, it has been referred to as Flammer syndrome, since it was first described by Katarina Konieczka and co-authors. There are two opposite extremes in society. Here, to the right, we have people with risk factors for atherosclerosis, such as obesity, arterial hypertension, diabetes, smoking, etc. And on the contrary, here to the left, we have people with risk factors for a Flammer syndrome, such as slimness, arterial hypertension, cold hands, etc. The people with risk factors for atherosclerosis are more likely to have an elevated intraocular pressure and thus have a slight increased risk for high-tension glaucoma. People with Flammer syndrome, however, are more likely to have a disturbed autoregulation and a low perfusion pressure, thus have a certain risk for normal tension glaucoma. Of course, this is a certain simplification. For example, there are people with FS who develop high blood pressure as they get older, or patients with normal tension glaucoma in whom the intraocular pressure increases with time, etc. Both groups discussed here share two other common characteristics, namely a tendency towards endothelial dysfunction, as shown in DVA, and elevated endothelin levels. This explains why both groups tend to have an increased retinal venous pressure. Both glaucoma and Flammer syndrome affect the retinal venous pressure, which is why the biggest difference between RVP and IOP is in the group of patients who have both. And this we find often in patients with normal tension glaucoma. Finally, we will briefly discuss a special clinical picture, namely the optic nerve compartment syndrome. In these patients, free communication of the cerebrospinal fluid is impeded at this location. One of the consequences is an increase in pressure within the compartment, especially after a Valsalva maneuver. It is therefore not surprising that the retinal venous pressure is also increased in the respective eyes. Interestingly, however, the retinal venous pressure often dropped only slightly after surgical decompression, but could then be reduced pharmacologically. This demonstrates that even in such situations, the increase in RVP is not only due to mechanical causes. This brings us to the treatment of an increased retinal venous pressure. As this is a young research area, the therapeutic choice is still limited. Nevertheless, we have some options that we will discuss now. We start with the calcium channel blockers. Endothelin increases calcium concentration in the cytoplasma, both by releasing calcium from the internal storages and by opening calcium channels. The CCBs inhibit only this second part. This explains why they weaken the endothelin effect but never completely cancel it out. Feng et al. Showed that while placebo had no effect, a low dose of nifedipine significantly reduced retinal venous pressure in glaucoma patients, especially in the group that had both glaucoma and Flammer syndrome. These authors here took a group of volunteers with a mountain railway at an altitude of 300 meters and observed an increase of RVP, which decreased again under therapy with an endothelin blocker. While this helps us to understand the regulation of the retinal venous pressure, it is unlikely that ET blockers will be used for this indication in the near future. Certain antioxidants also reduce RVP, as this example with porridge tea shows. This can be explained by the fact that such types of antioxidants slightly increase nitric oxide and lower endothelin. Nitrites, which we know from the treatment of angina pectoris, also lower RVP. Nitrites set NO3 in our body and indirectly also lower endothelin. Particularly interesting for us is orcofolin. It is a vitamin preparation which contains L-methylfolate, the active bioavailable form of folic acid. Beside an antioxidant effect, it also has a strong homocysteine-reducing effect. Several ophthalmologists independently observed that orcolin also reduces the retinal venous pressure very efficiently. Dr. Devogolaire in Belgium has studied this systematically. He shows these results in his presentation. This brings me to the end. I hope I could convince you that measuring and treating retinal venous pressure is scientifically interesting, clinically important, and helpful for patients. Thank you and goodbye.